human coronaviruses, first characterized in the 1960s, are responsible for a substantial proportion of upper respiratory tract infections in children. Since 2003, at least 5 new human coronaviruses have been identified, including the severe acute respiratory syndrome coronavirus, which caused significant morbidity and mortality. NL63 represents New Haven and NL coronaviruses. The history of human coronaviruses began in 1965 when Tyrrell and Bino found that they could passage a virus named B814. It was found in human embryonic trachean organ cultures obtained from the respiratory tract of an adult with a common cold. Almeida and Tyrrell performed electron microscopy on fluids from organ cultures infected with B814 and found particles that resembled the virus. These particles were medium-sized 80 to 150 nanometer, pleomorphic, membrane-coated, and covered with widely spaced, club-shaped surface projections. This new group of viruses was named coronavirus, corona denoting the crown-like appearance of the surface projections, and was later officially accepted as a new genus of viruses. Tyrrell was leading a group of virologists working with human strains. It was found that in temperate climates, respiratory coronavirus infections occur more in the winter and spring than in the summer and fall. Data revealed that coronavirus infections contribute as much as 35% of the total respiratory viral activity during epidemics. While research was proceeding to explore the pathogenicity, the number and importance of animal coronaviruses were growing rapidly. The newly identified Group 1 human coronaviruses were sequenced using a novel genomic amplification technique. These investigators were able to sequence the entire viral genome. The technique was RT-PCR, primed reverse transcriptase polymerase chain reaction, and it was used to identify the viral sequences. The propagation depended on cell culture. With the use of molecular probes that targeted conserved regions of the coronavirus genome, months later, Esper found evidence of a human respiratory coronavirus in respiratory specimens.
Taxonomy of coronavirus. Coronavirus are named for crown-like spikes on the surface. As experts on coronavirus, the ICTV Coronaviridae study group has studied the taxonomy of new virus, which is responsible for developing the classification of virus and taxon nomenclature of the family. Coronavirus belong to order Nidovirales and the family Coronaviridae. The Coronaviridae family gets its name in part because the virus surface is surrounded by a ring of projections that appear like a solar corona when viewed through an electron microscope. The family is divided into two subfamilies, Coronavirinae and Torovirinae, 4 genera, 26 subgenera, and 46 species. Still, some additional species are pending. Now, let's move on to the classification of coronavirus. Coronavirus are the members of subfamily Orthocoronavirinae. There are 4 main subgrouping of coronavirus such as alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. Human coronavirus were first identified in the mid-1960s. The 7 coronavirus that can infect people are 229E, NL63, OC43, HKU1. MERS coronavirus that causes Middle East respiratory syndrome. SARS coronavirus that causes severe acute respiratory syndrome. SARS coronavirus 2, the novel coronavirus that causes coronavirus disease 2019 or COVID-19. People around the world commonly get infected with these human coronavirus. Using molecular clock analysis, investigators have estimated the most common ancestor of all coronavirus appeared in about 8100 BC, and those of alpha, beta, gamma, and delta coronavirus appeared in approximately 2400 BC, 3300 BC, 2800 BC, and 3000 BC respectively. These investigators posit that birds and bats are ideal hosts for coronavirus gene source, bats for alpha and beta coronavirus and birds for gamma and delta coronavirus.
The coronavirus is spread through respiratory droplets that are produced when a person coughs, talks, or sneezes. When inhaled, the virus settles in the nasal passages and starts replicating throughout the body. It takes over the respiratory system's healthy cells along the way. If the virus enters the lungs, it can invade tiny air sacs or alveoli and causes pneumonia. At this point, the lungs will become inflamed and it's harder to breathe. In a CT scan, the lungs will have white hazy patches inside them. This is known as ground glass opacity. Acute respiratory distress syndrome, which is abbreviated as ARDS, sets in a few days later. The coronavirus and ARDS don't just pass on their own, they can cause significant lung damage or worse. If you are having difficulty in breathing, seek medical attention immediately.
Severe acute respiratory syndrome SARS was first recognized during a global outbreak of severe pneumonia that first occurred in late 2002 in Guangdong province, China, and then erupted in February 2003 with cases in more than 24 countries in Asia, Europe, North America, and South America. The disease causes an influenza-like illness, and in severe cases it can cause human death. Person-to-person transmission combined with international travel of infected persons accelerated the worldwide spread of the illness, and by the time of the outbreak was contained, 8,998 probable cases resulting in death of 774 people were reported, resulting in the first human pandemic of 21st century. A global network of 11 laboratories was established by the World Health Organization to identify the causal agent. A novel coronavirus was identified as the etiological agent of SARS. This virus belongs to a family of large positive single-stranded RNA viruses. Genomic characterizations showed that SARS-CoV is only moderately related to other known coronaviruses. SARS-CoV infection typically causes severe symptoms related to the lower respiratory tract. The virus has been isolated from several animals including civet cats and raccoon dogs. Recently, certain bat species have been reported as potential natural reservoirs. SARS has a characteristic clinical course. Patients present with flu-like symptoms including fever, chills, cough, and shortness of breath, and recurrent of persistent fever. Approximately 20 to 30% of patients requires intensive care treatment, including mechanical ventilation. Several reports have described diffuse alveolar damage with various levels of progression and severity as the main histopathological findings in SARS patients. Lungs typically shows changes in proliferative phase of diffuse alveolar damage, with hyaline membrane formation, disformation of epithelial cells, fibrin deposit in the alveolar space, and hyperplasia of type 2 pneumocytes. Mononuclear infiltrate in the interstitium can be seen in some cases. Other findings identified in some patients included focal intraalveolar hemorrhage, necrotic inflammatory debris in small airways, and organizing pneumonia. In addition, multinucleated syncytial cells were seen in the intraalveolar space of some patients who died in 14 days or more after onset of illness. These cells contain abundant vacuolated cytoplasm with cleaved and convoluted nuclei. No obvious intranuclear or intracytoplasmic viral inclusions were identified. In many cases, cellular infiltration has been observed. Immunohistochemical staining has shown that these inflammatory cells predominantly consist of macrophages. Certain studies have compared as the pulmonary pathology of SARS cases with non-SARS cases showing SARS-like symptoms. Therefore, additional tests such as in situ hybridization, IHC, viral isolation, or RT-PCR are necessary to confirm the diagnosis. In most SARS autopsies, both extensive necrosis of the spleen and atrophy of the white pulp with severe lymphocytes depletion have been found. Lymph nodes usually show atrophy and reduction of lymphocytes with loss of germinal centers. Lymph nodes usually show atrophy and reduction of lymphocytes with loss of germinal centers. Kidneys of autopsied SARS patients have shown focal necrosis and vasculitis of small veins in the renal interstitial tissue. In addition, monocytes infiltration and acute tubular necrosis and other nonspecific changes such as glomerular fibrosis and sclerosis have all been observed. Gastrointestinal manifestations are commonly reported in SARS cases, with more than 20% of patients presenting with watery diarrhea and up to 67% of patients developing diarrhea during the course of the illness with inflammation around both intestines. The pancreas, stomach, and pituitary glands have not shown in obvious pathological changes. The majority of SARS patients show an increase in serum alanine aminotransferase levels during the course of their disease. High peak alanine aminotransferase levels has been associated with an adverse outcome. In some autopsy cases, generation necrosis of hepatocytes and cellular infiltration were observed. In some cases, evidence of reactive cytosis or bone marrow hypoplasia was present. Destruction of epithelial cells with significant changes in the follicular architecture was present in the thyroid glands. The few available studies on adrenal glands describe the presence of necrosis and vasculitis of the medulla with monocytes and lymphocytes. Infiltration viral agents and genomic sequence have been identified in adrenal glands. Mechanisms contributing to pathogenesis of SARS. These pathological events and cascade of changes form the basis for clinical symptoms and pathological findings at different stages of SARS. Correct understanding of the pathogenesis will provide guidance to prevention, diagnosis, and treatment of this new disease.
We will deal with the life cycle of coronavirus. Virion attaches to the host cell by a specific protein on its surface called spikes. Spike protein S1 and S2 of SARS-CoV-2 mediate attachment to the membrane of a host cell and engage angiotensin-converting enzyme ACE2 as a receptor. These spike proteins are activated by a cellular serine protease TMPR22, which are close to ACE2 receptor, aid in viral entry. Enveloped virus fuses with the plasma membrane of the host and nucleocapsid is directly released into the cytoplasm. Nucleic acid then separates from the protein coat. The host ribosomes translates the viral RNA from the 5' end. From the 5' end of the positive strand viral RNA, first to be read is a leader sequence which enables the proper alignment of the host ribosome, followed by 5' untranslated region which regulates viral translation. First key protein to be produced is RNA-dependent RNA polymerase responsible for viral replication. Then a set of 3 structural proteins are translated, of which first protein is S protein, which in its glycosylated form. The protein formed during this stage is inactive. Second protein to be synthesized is enveloped protein, which is essential for viral assembly and release. Third protein to be synthesized is a membrane protein, which imparts circular curved shape to the virus. It binds to the nucleocapsid protein. All the structural proteins are transported to the lumen of endoplasmic reticulum, where they are glycosylated. Then they are transported to the Golgi apparatus through vesicular transport. On maturation, structural proteins assemble and polymerize to form a nucleocapsid and stay inside the Golgi bodies until they receive replicated viral RNA. Ribosomes now translate the nucleocapsid protein, which helps replicate transcriptase complexation as well as assist the viral RNA packaging by binding viral genome in specific conformation. The ribosomes now glide to 3' untranslated region, which contains a pseudo-knot structure which is essential for viral genome replication. Before disintegrating, ribosomes glide over to the poly A tail, which protects the viral RNA from enzymatic degradation in host cell, while also signaling the end of translation. RNA polymerase reads the RNA from the 3' end to the 5' position and synthesize a complementary strand in the anti-parallel direction from 5' to 3'. From the first cycle of positive strand RNA replication, a negative RNA strand is formed, which serves as a template. This negative strand RNA is not used for translation nor transcription. This negative RNA strand only serves as a template to produce more positive RNA strands. The complementary strand from the negative RNA strand will now bear a same sequence as the original positive RNA strand. Therefore, this RNA is used for translation and packaging. However, a negative RNA strand will remain in the cytoplasm of the host cell to produce more copies of the positive strand RNA. The positive RNA strand will bind with the nucleocapsid protein and transported to the Golgi apparatus and is packed into viral particles. The mature viral particle escape from the Golgi apparatus in an entrapped form inside the vesicle. These vesicles containing viral particles will travel to the periphery of the host cell, where they can facilitate the exocytosis of the viral particles. The virus particle infects the new healthy cells.
COVID-19 Coronavirus Disease 2019. COVID-19 is a disease caused by a new strain of coronavirus. CO stands for corona, VI stands for virus, D stands for disease. Formerly, this disease was referred to as 2019 novel coronavirus or 2019 nCoV. In early 2020, after a December 2019 outbreak in China, the World Health Organization identified SARS coronavirus 2 as a new type of coronavirus. The outbreak quickly spread around the world. COVID-19 is a disease caused by SARS coronavirus 2 that can trigger what doctors call a respiratory tract infection. It can affect your upper respiratory tract, sinuses, nose and throat, or lower respiratory tract, windpipe and lungs. It spreads the same way other coronaviruses do, mainly through person-to-person contact. Infections range from mild to deadly. SARS coronavirus 2 is one of 7 types of coronavirus, including the ones that cause severe diseases like sudden acute respiratory syndrome, SARS. Strains of COVID-19. The virus causing the COVID-19 pandemic, SARS coronavirus 2, presents at least 6 strains. Despite its mutations, the virus shows its little variability. Currently, there are 6 strains. The original one is the L strain that appeared in Wuhan in December 2019. Its first mutation, the S strain, appeared at the beginning of 2020, while since mid-January 2020 we have had strains V and G. To date, strain G is the most widespread. It mutated into strains GR and GH at the end of February 2020. GR strain is reported in Europe, Italy, South Africa, Asia. GH strain is reported in France, Germany, North America, Asia. G strain is mostly reported in Asia, whereas S strain is mostly reported in US and Spain. Besides these 6 main coronavirus strains, researchers identified some infrequent mutations that at the moment are not worrying but should nevertheless be monitored. The main symptoms include fever, coughing, shortness of breath, trouble breathing, fatigue, chills, sometimes with shaking, body aches, headache, sore throat, loss of smell or taste, nausea, and diarrhea. If a person is infected, symptoms can show up in as few as 2 days or as many as 14 days. It varies from person to person. These symptoms are common, but then how is COVID-19 different from other diseases? And why is COVID-19 life-threatening? There are many adverse effects of COVID-19 disease and is life-threatening, which creates a difference between other diseases and COVID. As the virus can lead to pneumonia, respiratory failure, septic shock, and death. Many COVID-19 complications may be caused by a condition known as cytokine release syndrome or a cytokine storm. This is when an infection triggers your immune system to flood your bloodstream with inflammatory proteins called cytokines. They can kill tissue and damage your organ. A severe lung condition that causes a low amount of oxygen to go through your bloodstream to your organs, acute respiratory distress syndrome, blood clots, and acute kidney injury. These adverse effects make COVID-19 a life-threatening disease, as these are the after effects of COVID-19, which is a threat to life. What is the age group target of COVID-19 and why? It can lead to hospitalization and even death for young and middle-aged adults. It has caused the most severe health issues for adults over the age of 60, with particularly fatal results for those 80 years and older. Additionally, as people age are more susceptible to infection of any kind, especially a new one like COVID-19. This is why COVID-19 is different from other diseases and is a pandemic.
Introduction on the structure of coronaviruses. Coronaviruses has a circular viral membrane composed of a lipid bilayer which possess transmembrane proteins. They are generally large, envelope positive-sense single-stranded RNA viruses. Structure of SARS coronavirus 2. SARS coronavirus 2 is a beta coronavirus. They are envelope positive-sense single-stranded RNA viruses. They are spherical teleomorphic particles measuring between 80 and 160 nanometer in length. It contains 4 structural proteins, namely envelope, spike, membrane, and nucleocapsid. The properties of each structural protein are as follows. Spike protein or S protein. It is involved in the formation of envelope of virus. It is also involved in virus assembly during replication. Spike protein attached to the human receptor protein, that is angiotensin converting enzyme 2. This S protein or spike protein is the one that appear crown-like under electron microscope, hence named as corona. The next protein is nucleocapsid protein. It is a ribonucleoprotein. It forms a complex with RNA, assist in viral assembly. E protein or envelope protein. It forms the viral envelope of virus. The next protein is M protein or membrane protein. It is the most abundant protein which is mostly responsible for the shape of the envelope, whereas the another protein is hemagglutinin esterase gene, which is a characteristically present in the beta coronaviruses group, but it is lack or absent in SARS coronavirus 2. Structure of spike glycoproteins. The detailed structure of spike protein was discovered by researchers by using cryo-electron which yield a detailed 3D view of a spike glycoprotein. Spike glycoprotein are curve shaped. It consists of two subunits that are subunit 1 and subunit 2. Subunit 1 consists of three segments that are a large ectodomain, a transmembrane, a receptor binding domain. S2 subunit consists of fusion peptide, cytoplasmic domain, transmembrane domain, heptad repeat 1 and 2. Hence, the spike protein of SARS coronavirus 2 subunit shares 70% similarity and subunit 2 shares 99% of similarity with SARS coronavirus. Genome organization of SARS coronavirus 2. Researchers rapidly discovered the genome sequence of SARS coronavirus 2. It was first released by the scientists of China to the public. According to the researchers, the genome of SARS coronavirus 2 is single-stranded positive RNA of 30 kb, 29,891 nucleotides encoding 9,860 amino acid. The guanine plus cytosine content is 38%. The genome is arranged in the order of non-coding 5' cap untranslated region and non-coding 3 cap UTR region. 5' UTR region contains several open reading frames that encode multiple proteins, and 3 terminus of SARS coronavirus 2 contain 4 structural proteins that are spike, envelope, nucleic acid, and membrane protein. 2/3 of viral RNA, mainly located in first ORF, translate to polyprotein PP1A and PP1AB and encode 16 non-structural proteins. 16. NSP include two viral cysteine proteases, namely NSP3 papain-like proteases and NSP5 main proteases, NSP12 RNA-dependent RNA polymerase, and NSP13 helicase. The rest part of genome code for 4 structural protein E, M, S, and N, along with a number of accessory protein. SARS coronavirus 2 is closer to SARS-like coronaviruses in the term of whole genome. Due to mutations in NSP2 and NSP3 and the spike protein, which play a significant role for the infections and different mechanisms of SARS coronavirus 2. How is SARS coronavirus 2 different from other coronaviruses? Despite high sequence similarity, a few notable and conserved variation has been arose in SARS coronavirus genome that are not seen in beta coronaviruses. Notable features which establish this virus as different from other coronavirus are as follow. Multiple mutation in receptor binding domain of a spike protein that may interact with angiotensin converting enzyme 2 receptor. Polybasic furin-like protease site at the boundary of S1 or S2 subunit rather than single arginine observed in SARS coronaviruses. Addition to this, the protease site which is present in the SARS coronavirus 2 is not present in beta coronaviruses. It is a signature of a highly pathogenic influenza species.
Moving on with our next discussion matter, post-COVID mutations, that is, how the virus has undergone mutation post its journey from China. Virus, at its most basic, is bunch of genetic material packed into a protein shell. It replicates inside its human host, uses us to multiply by injecting its genetic information into our cells. The cell then becomes a kind of virus factory, mass producing the genetic material that allows more of the virus to develop. As it replicates, mistakes are often made. Scientists call these mistakes mutations and have been tracking those mutations following coronavirus all over the world. A mutation is a change in a DNA sequence. They can result from DNA copying mistakes made during cell division, exposure to ionizing radiation, exposure to chemicals called mutagens, or infection by viruses. As genetic code is designed to be conservative, most mutations don't actually change how virus behaves in our body. They are a part of the process and slowly go away. Back when researchers first analyzed the genome of a brand new coronavirus in Wuhan, its genetic sequence looked like this. 3 weeks later, same viral genome was sequenced again. It had changed. Coronavirus was mutating. So, the question of the hour is coronavirus becoming more dangerous? That is, if it causes a more severe disease or if it is more contagious? When COVID 19 spread around the globe this year, David Montefiore wondered how the deadly virus behind the pandemic might be changing as it passed from person to person. Montefiore is a virologist who has spent much of his career studying how chance mutations in HIV help it to evade the immune system. The same thing might happen with SARS CoV 2. There are two conformations of virus, closed and open. These conformations are of the spike protein or SARS-CoV-2, which binds to receptors on human cells. A common mutation seems to make the protein favor open conformations, which might mean the virus can enter cells more easily. It had already started scoring thousands of coronavirus genetic sequences for mutations that might have changed the virus's properties as it made its way around the world. All these mutations happen during a process called replication. This replication process is messy. Viruses that encode their genome in RNA, such as SARS CoV 2, HIV, and influenza, tend to pick up mutations quickly as they are copied inside their hosts. Because enzymes that copy RNA are prone to making errors. Even though it has a proofreading mechanism for copying its genome, the coronavirus frequently makes mistakes leading to mutations. Compared with HIV, SARS CoV 2 is changing much more slowly as it spreads, but scientists are concerned about one mutation in particular that might have impact on how the virus spreads. It was in the gene encoding the spike protein, which helps virus particles to penetrate cells. The red protein that everyone sees on surface of coronavirus, there is more of that on mutated viruses. At the 614th amino acid position of the spike protein, the amino acid aspartate D in biochemical shorthand was regularly being replaced by glycine because of a copying fault that altered a single nucleotide in the virus's 29903 letter RNA code. Virologists were calling it the D614G mutation. This global shift of the SARS-CoV-2 variant is from D614 to G614. The G614 variant is more infectious in laboratory settings. Whether it means increased viral spread in humans is unconfirmed. Current evidence says that the G614 variant is not any deadlier than D614. And so, treatment options should not be any more different. Both the D614 and G614 variants should react similarly to vaccines, as the mutation does not change the immunogenic part of the spike protein, that is, the receptor binding domain. G614 had rapidly become the dominant SARS CoV 2 lineage in Europe and had then taken hold in the United States, Canada, and Australia. D614G mutation represented a more transmissible form of SARS CoV 2, one that had emerged as a product of natural selection. One major change in virus has already been determined. Earlier generations of coronavirus replicated in the lungs. A new mutation using host cells somewhere else in a body, that is the throat, is more contagious if not more deadly. Most available evidence suggests that D614G doesn't stop the immune system's neutralizing antibodies from recognizing SARS CoV 2 as Montefiore had worried. That might be because the mutation is not in the spike protein's receptor binding domain. So, to conclude, I want to say mutations happen. They are a part of natural rhythm of a virus. They are not inherently good or bad. Scientists are watching them closely, but they are not expecting a science fictionary monster movie scenario. It is still unknown whether this small mutation affects the severity of symptoms of infected people or increases mortality. Let's hope for the best in future.
Change in clinical course of infection of COVID-19 from pulmonary to cardiac. 80% of the cases are mild to moderate, 14% are severe, and 6% are critical. A mild case of COVID-19 is not like a mild cold. The symptoms still be pretty severe. It may take 2 or 14 days to develop the first symptom. Clinical observations suggest that platelets serve important functions during COVID-19 progression. Due to the abundance lack of nucleate, platelets are more likely to get stick to the virus particle. Platelets perform various functions in concern with neutrophils, erythrocytes, and endothelial cells, which can increase the cumulative incidence of thrombosis. Multiple mechanisms may be contributing to thrombosis, like increase in production of platelets, increase in fibrinogen, by neutrophils, and injury to the endothelial cells. Human platelets, the initial response to viral RNA is mediated by toll-like receptor 7. TLR7 leads to complement C3 release. C3-mediated netosis occurs. Netosis is a type of programmed cell death where neutrophil extracellular traps capture and protect from virus, but are also highly protothrombotic, and when they are dysregulated may induce intravascular coagulation. Thrombin, which is generated from coagulation, can in turn activate C3 and the entire pro-inflammatory cascade. Uncontrolled netosis can cause severe cell and organ damage. In addition to C3, platelets also secrete granulocyte macrophage colony stimulating factor (CMCSF), which controls the level of DNA released from neutrophils. The damage caused by inflammation and netosis during infection can lead to platelet tissue factor release. Tissue factor would further increase by thrombotic response of platelets by generating thrombin and fibrin fibers. Lack of functional endothelium and tissue factor contribute to the increased tendency for thrombosis in small vessels. COVID-19 infection is accompanied by an aggressive inflammatory response with the release of large amount of pro-inflammatory cytokines. This is known as cytokine storm. Immune response of human body to SARS-CoV-2 is hyperactive, resulting in an excessive inflammatory reaction. Cytokines are produced by several immune cells, including macrophages, dendritic cells, natural killer cells, adaptive T and B lymphocytes. During an innate immune response, pattern recognition receptors recognize pathogen-associated molecular patterns (PAMs). Binding of PAMs to PRRs triggers the start of inflammatory response, resulting in activation of several signaling pathways and transcription factors. Three most important cytokines of innate immune response are interleukin-1, interleukin-6, and tumor necrosis factor alpha. The increase in cytokines results in influx of macrophages, neutrophils, T cells near site of infection, resulting in destabilization of endothelial cell-to-cell interaction, which causes capillary damage. Lung injury is one of the consequences of cytokine storm, which can progress into acute lung injury or more severe form, that is, acute respiratory distress syndrome. Infected endothelium, particularly in small vessels, can lead to thrombosis. Direct infection of endothelial cells may lead to damage of endothelial lining by apoptosis or pyroptosis. As more endothelial cells become infected, more platelets will aggregate and form microthrombi in the vessels. Additionally, platelets can react to inflamed and infected endothelium, inflammatory cytokines, and the antibody level changes, which can all contribute to the amplification of thrombotic function. As thrombocytopenia and microthrombosis are difficult to resolve, so targeting inflammatory cytokine signaling and netosis during COVID-19 may be a better approach than providing antiplatelet drugs. The identification of the most relevant intracellular signaling pathways involved in the modulation of host immune response may give us important hints to overcome the disease. Toll-like receptors have been found to activate different signaling pathways in human CD14 cells, monocytes, interferons, and cytokine secretions. Downstream transduction pathways which are crucial for proper antiviral response are interferon regulatory factor 3, nuclear factor, JAK-STAT signaling pathways. Cytokine interleukin-6 is found in COVID-19 as a massive cytokine release. Interleukin-6 is able to activate a soluble form of interleukin-6 receptor interacting with glycoprotein 130, thereby promoting the downstream activation of JAK-STAT signaling. The activation of JAK-STAT pathway in turn stimulates the production of interleukin-6. Abnormal increase in interleukin-6 levels interferes with the cause of cardiovascular diseases like coronary arteriosclerosis, microangiopathy with thrombosis. Interleukin-6 production is also induced by angiotensin II in AT1 JAK-STAT dependent manner. Angiotensin II is a peptide hormone. SARS-CoV-2 downregulates ACE2 receptor expression, which results in overproduction of angiotensin II, which increases vascular and lung injuries.
Serology and diagnosis of COVID-19. In response to COVID-19 pandemic, multiple diagnostic test manufacturers have developed and began selling rapid and easy-to-use kits. Before tests are put in use, they must be validated. Inadequate tests may miss patients with active infection and may falsely categorize people as having the disease, further hampering disease control efforts. Two terms which are commonly used in diagnostic tests are specificity, that is, the percentage of healthy people as not having the condition, and sensitivity, which is the number of sick people correctly identified as having the disease. The two main branches which detect either the presence of virus, which is known as the viral tests, or the presence of antibodies produced in response to the infection, which is known as the antibody tests. Tests for viral presence are used to diagnose individual cases and to allow public health authorities to trace and contain outbreaks. Antibody tests show whether previously the person had the infection and are less useful for diagnosis. Each diagnostic test targets a specific viral component of the virus to aid in its detection. The first molecular test for viral detection is RT-PCR, which is also the gold standard for testing of COVID-19. It amplifies genetic material many hundreds, thousands of times, creating enough for analysis. The process uses reverse transcriptase to obtain DNA from viral RNA. PCR targets the envelope protein, RNA-dependent RNA polymerase, and also the nucleocapsid protein. Fluorescent dyes are used to visually depict the result. Next is isothermal amplification assay, which amplifies the genome and is faster than PCR. It utilizes CRISPR gene editing technology, which was modified to perform the detection. This test detects the viral genome with the help of fluorescent tags, which are read out using specialized machines. An added benefit is that it amplifies RNA directly without the need to convert it into DNA. Next are the antibody tests. These can be used to assess the fraction of population which was once infected with the virus. The most notable antibodies produced are IgG and IgM. IgG is produced after 10 to 14 days of initial infection. Rapid diagnostic test utilizes a conjugate pad which has reagents for optimized reaction. Results are depicted by colored particles such as nano-sized gold particles or fluorescence. Next is ELISA. It has a plate which is coated with viral proteins such as the spike protein. When the patient sample is added, the antibodies bind to it, forming a complex. This can be detected by another wash of antibodies that produce color or are fluorescently tagged. Other assays include neutralization assay and chemiluminescent assays which follow the same principle. Furthermore, imaging can be used to confirm results of COVID-19 infection. Visible features such as ground glass opacities develop as the disease evolves. Hence, it is necessary to get yourself checked if any of the symptoms are seen. The various samples collected are nasal swab, throat swab, saliva, sputum sample, tracheal aspirate, or even a blood test. It is very necessary to follow strict guidelines and protocol for processing and handling the samples. Healthcare professionals must wear a PPE kit to ensure safety. The sample must be collected in an aseptic manner to avoid contamination. It is important to store the sample collected in an appropriate medium such as viral transfer medium. Worldwide tests available are mostly nucleic acid tests. China, France, Germany, Hong Kong, Japan, UK, US all targeted different parts of the viral genome. PowerCheck coronavirus looks for the E gene shared by all beta coronaviruses and the RDRP gene specific to SARS-CoV-2. About Laboratories kit amplifies RDRP gene, resulting copies are detected with fluorescently labeled molecular beacons. Now about India's testing strategies. Testing had been initially restricted to
high-risk individuals, leading to a biased testing sample, which led to missing the actual nature of the outbreak. India, being a largely populated country, has a disadvantage at hand. India needs to drastically expand testing randomly, including community spread and asymptomatic cases. This will help understand the transmission characters and plan appropriately for the future. India's first CRISPR COVID-19 test, launched by Tata & Sons Feluda, uses CRISPR technology to detect the genome of SARS-CoV-2.
Here is the vaccine trials in phase 1, phase 2 and phase 3. Only 5 is showing results, but approved is 0. Bharat Biotech, in collaboration with the Indian Council of Medical Research and the National Institute of Virology. The Indian company Bharat Biotech designed a vaccine called Covaxin based on an inactivated form of the coronavirus. The company launched phase 1 and phase 2 trials in July. In India, Zydus Candela began testing a DNA-based vaccine in July. They launched a phase 2 trials on August 16. It is the second company in India to enter a COVID-19 vaccine race after Bharat Biotech. Moderna, in partnership with the National Institutes of Health, they found the vaccine effective against the coronavirus. In US, the Moderna developing a vaccine based on mRNA to produce viral proteins in the body. In March, the company put the first COVID-19 vaccine into human trials, showing promising results. The vaccine has progressed in phase 3 testing on July 27. The final trial is 30,000 healthy people at about 89 sites around the US. It is a mRNA-based vaccine, and the vaccine name is mRNA 1273. On September 17, Moderna shared their protocol for their vaccine. The vaccine may take till at the end of 2020 or early 2021 to reach the world. I am sharing a Moderna's protocol how the vaccine they are preparing. The trial volunteers will receive 2 intramuscular injections approximately 20 days apart. Participants will receive 2 injections of 100 microgram of vaccine mRNA 1273 or shots of saline placebo. The spike protein on virus surface facilitates its entry into a cell. Moderna's mRNA 1273 uses messenger RNA that encode for an S2P immunogen. The vaccine directs the blood cells to express the spike protein to get a broad immune responses. Volunteers must have to provide nasopharyngeal swab and a blood sample at an initial screening. It is a New York-based vaccine. The vaccine containing parts of several viral proteins. On September 11, they registered a phase 1 trial in Taiwan. The Australian company Vaccine developed a vaccine that combines viral proteins with an adjuvant that stimulates the immune system. They successfully completed phase 1 trials in July and expect to start phase 2 trials in September. Here is another private Chinese company, Sinovac Biotech, is testing an inactivated vaccine called Coronavac. In July, the company announced that phase 1 phase 2 trials on 743 volunteers found no severe adverse effect and produced an immune response. They launched a phase 3 trial in Brazil in July and another in Indonesia in the following month. The Chinese government gave the Sinovac vaccine an emergency approval for limited use in July. There are so many trials are going on coronavirus vaccine. Some of them are listed below.